MCAT Organic Chemistry. We're going to take a look at some different types of alkanol reactions, starting with dehydration, which we can call elimination. Now, with dehydration, the products formed depend on the conditions used. For example, alkenes are formed in the presence of sulfuric acid. Now, phosphoric acid is better since it does not produce as many byproducts. The correct temperature is also important because hot is used for primary, warm for secondary, and cool for tertiary. And then alcohols actually lose a water molecule in the process of dehydration. Now, with oxidation, the products depend on the type of alcohol used: primary, secondary, or tertiary. For primary and secondary, a double bond between carbon and oxygen replaces a single bond between carbon and hydroxide. But this bond will either be at the end of the carbon chain, which would make it an aldehyde, or in the middle of the carbon chain, making it a ketone. Aldehydes can be further oxidized to form carboxylic acids, and tertiary alcohols will not oxidize. Now we come to substitution, which is usually not very important for the alcohols except for halogenation using phosphorus pentachloride. So substitution may be used as an identifying test for the alcohol group. Then a reaction with active metals is just what it sounds like. When alcohols react with active metals, and in that they release hydrogen. And so this also may be used as an identifying test for alcohols. Now, esterification is a reaction between an alcohol and a carboxylic acid. And this forms an ester link, which looks like this. And this holds two carbon chains together. Now, the conditions required for this reaction are concentrated sulfuric acid and elevated temperatures.
Antibodies are also known as immunoglobulins. And they are large Y-shaped proteins produced by B cells. Now, an antibody is used by the immune system. You might have been clued in by the immunoglobulin alternative name. So it's used by the immune system to identify and neutralize foreign objects such as viruses and bacteria. So if you inhale or ingest something that is foreign to your system, antibodies are going to kick in and try to neutralize these foreign objects. The antibody recognizes a unique part of the foreign object called an antigen. So it's important to remember the difference between these two, since antigen and antibody are both similar to each other. So the antigen is what's part of the immune system and is going to stop the foreign objects. The antigen is a specific part of that foreign object that we're going to try to neutralize. Each tip of the Y, remember we said an antibody is a Y-shaped protein. Each tip of the Y has a paratope. And think of the structure of a lock for a paratope. And each paratope will only bind to a specific epitope. And think of it like a key. So let's look at our structure over here. We've got our antibody, which is this whole Y-shaped protein. And then we've got all these antigens up here. So there are lots of different antigens that could come into your body, and this is probably not what they look like, but it's just to give you an example. If we're thinking of our antigens, and this area here is going to be our epitope, this part here that would bind, it's supposed to be considered a lock. So if this, it's supposed to be considered our key. So if this is our lock here, this antigen binding site, or the paratope, then it's only going to fit with one key. A lock is only going to be unlocked with one key. You have to have the right key to get it to unlock. So we've got our antibody, and each part of the Y is only going to bind to a specific antigen. The paratope, this little shape on the end of our antibody, is only going to bind to one epitope, which is located on an antigen. So that one spot on your antigen is what's going to be able to bind to the one spot on the antibody. And if you don't have the right key to go in the lock or the right lock to bind with the key, then nothing's going to happen. So there are lots of antigens, and different antibodies can attach to different ones of those. But one antibody is only going to be able to bind to one antigen. If you have lots of antibodies floating around in your body, each one is going to only be able to attach to one antigen. So you may have lots of them that can attach to one antigen and lots that can attach to another, but you can't just say, okay, well, I don't really have this antigen in my body, I have this one, so let me plug that in there, because it's not going to fit. You have to think of it like a lock and key. So once you have this antibody produced, it's only going to be able to bind to the one antigen to neutralize it. So once the antigen and antibody bind, and remember, special epitope-paratope relationship, lock and key, it's only going to be able to bind to one specific type of antigen. Once they bind, the foreign object is tagged for attack by another part of the immune system, or it's neutralized directly. There are some antigens, some foreign objects where if we block the antigen, then that foreign object isn't going to be able to do any more harm in your body. So the antibody can neutralize it directly. But there are other foreign objects that can't be neutralized quite so easily. So instead, there is something secreted by the antibody, and it coats the foreign object, and then the foreign object is tagged for attack, and some other part of the immune system steps in and removes the foreign object. So now that you've seen your antibody and seen how it binds to antigens and how it can either get rid of or neutralize the foreign objects directly or alert the attack for another part of the immune system, let's go back to the B cells. Remember, antibodies are produced by B cells. So once a B cell has been activated, once there's some foreign object in your body that the B cells know about and they say, okay, we've got to do something now, we've got to get rid of this. The B cells can turn into antibody producing cells called plasma cells. So they can produce new antibodies, these plasma cells that the B cells turned into, or they can be turned into memory cells that can remember an antigen and respond faster in the case of future exposure. So these B cells can turn into plasma cells that produce more antibodies if needed, or they turn into memory cells. And the memory cells are going to remember the last antigen they had, and they're going to be able to respond faster. So once this antibody has attached to this antigen, it's going to remember it next time, and it's going to be able to respond faster. It won't have to try all of these keys to figure out which one goes in the lock. It will already know which key goes in the lock, and it will be able to respond faster. So that's why if you've been sick with something before, you might not get it again, like chicken pox. Once you've had it, you usually don't get it again. Let's see. You get a flu shot and it's got antibodies in it. Once your body learns how to attack that flu virus, at least that particular strain of flu virus, then it will be able to attack it if it comes into your body again. Now, different strains of flu virus are going to have different antigens, so it won't be able to attack every flu virus, only the ones that have that same antigen that your flu vaccine had in it. So antibodies are very helpful with maintaining your immune system and keeping you healthy, or keeping any organism healthy. And the B cells produce the antibodies, The antibodies find foreign objects, find the antigen on them, and the particular epitope that it can bind to. And once they bind, that foreign object is tagged for removal. It's either going to be attacked by another part of the immune system, or it's going to be neutralized directly and it won't be able to harm anything and will eventually get transferred out of the body. And you've got your plasma cells that are going to produce more antibodies for your body, and your memory cells that are going to remember past antigens, past foreign objects that have come into your body, and know how to respond to them faster in the future. So remember, antibodies are also called immunoglobulins, and that will help you remember it's a part of the immune system. Remember the difference between antibody and antigen. The antibody is what is fighting the foreign object. The antigen is that specific part on the foreign object that has to be bound to be able to attack that foreign object and get rid of it. Aerobic respiration. Let's look over here first. In aerobic respiration, step 1 is always glycolysis. And during glycolysis, you have 2 pyruvate and 2 ATP molecules that are formed. So that gives you a little bit of energy and the pyruvate to start the rest of the aerobic respiration process. So step 1, glycolysis, where you get the pyruvate and the energy that you need to keep going. So the next step is where our pyruvate is converted into 2 acetyl groups that are combined with coenzyme A to form acetyl-CoA. And that's just an abbreviation letting you know it's the acetyl groups combined with coenzyme A. And we got this from pyruvate, which we got from glycolysis, where some glucose was broken down. So let's keep going. Next, the acetyl-CoA enters the matrix of the mitochondria, where it enters the Krebs cycle. And this is also called the citric acid cycle. So we've got our acetyl-CoA entering the matrix of the mitochondria. This is where it's going to take place. And then it enters the Krebs cycle. During the Krebs cycle, 8 additional hydrogen-rich NADH molecules and 2 additional FADH molecules are generated during the Krebs cycle. So we've put our pyruvate, we've broken down our pyruvate into 2 acetyl groups, combined them with coenzyme A. We've turned that acetyl-CoA through the Krebs cycle into these 8 additional NADH hydrogen-rich molecules and the 2 FADH molecules. And now we're going to keep moving them. And in the next step, these are reduced. That's going to be in the electron transport chain, still in the mitochondria. So, electron transport chain, these are reduced, and the resulting electrons create a proton gradient. And these protons that have been created kind of cascade down the gradient, and as they do that, in ATP synthase, they generate 30 or more molecules of ATP. So just imagine those protons cascading down, and as they slowly fall down, they're generating energy, and it can be 30 or more molecules of ATP that are generated. So let's look at all this in a diagram, maybe simplify it a little. So we've got our cell, and nutrients are taken into that cell. In the nutrients, we're going to get some glucose. And from 1 molecule of glucose, we are going to carry out glycolysis and get 2 ATP net energy over here, plus our pyruvate. And all of this happened in the cytoplasm of the cell. So we're inside the cell, but we're in the cytoplasm. But next, the pyruvate is going to go into the mitochondria. Singular mitochondrion. So it's just going in this one, and it's going to keep going, and the acetyl-CoA that we got from the pyruvate is going to go into the matrix of the mitochondria and enter the Krebs cycle. During the Krebs cycle, we're going to get these molecules we talked about, our hydrogen-rich NADH and our FADH, and they're going to enter the electron transport chain. And as they are reduced, they form that proton gradient. The protons cascade down and generate 30 or more ATP molecules with some waste particles in the form of H2O, water, and carbon dioxide. So there are some waste products here, but oxygen went in because this is aerobic respiration. We got 30 plus molecules of ATP plus the 2 from glycolysis, so a net of 32 total. Now that could differ because the mitochondria has this membrane around it, and some energy can escape from the membrane and some energy can escape from the cell. So sometimes you lose some of that energy along the way, but on average you could get 30 to 32 net ATP molecules from one molecule of glucose. And then you do have some waste products left over. This is a really complicated process. Hopefully this has simplified it for you, but the primary reason that a cell is going to undergo aerobic respiration is so that it can get a lot of energy and expel some waste products. DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curved shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat. And it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine thymine pair or guanine cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Enzymes. Enzymes are highly selective catalysts that greatly accelerate the rate of reactions. So they speed up how quickly a reaction takes place. Most enzymes are proteins, but some catalytic RNA molecules have been discovered. So there are RNA molecules that will catalyze reactions and help speed them up in the same way that these proteins will. And so they both work as enzymes. In enzymatic reactions, the molecules at the beginning of the process are known as substrates. During the reaction, these substrates are turned into different molecules known as products. And at the end of the reaction, the enzyme will be unchanged. The enzyme may have to change shape a little bit to bind to the substrate and finish the reaction, but the enzyme itself will be unchanged. It will not have turned into different products. It will not have gotten bigger or smaller. It will remain unchanged. Enzymes work by lowering the activation energy of a reaction, drastically reducing the reaction time. If you had to build up all that energy to complete a reaction normally, it would take a really long time. Well, whenever the enzyme works on a reaction, it lowers the activation energy and makes the reaction happen a lot faster. In fact, a lot of our metabolic processes would not be fast enough to sustain life without enzymes there to speed up the reactions. Enzymes are highly specific for their substrates. That means that each enzyme will only bind with specific substrates that fit with its active site. So each enzyme has an active site. Here we've got our enzyme. To give you an example, this area would be the active site. So both here and here, this open area in the overall shape of the enzyme is the active site, and each enzyme has an active site, and only substrates with a similar shape that would be able to fit in there and bind without the enzyme changing shape too much will be able to fit with that enzyme and complete a reaction. So this would be your substrate. And this is where the substrate would be coming into contact with the enzyme. Here you can see the enzyme and substrate have bonded, and now they have formed the enzyme-substrate complex. And you can see that the enzyme changed shape a little bit to bind to that substrate. And then in this next phase, you have the enzyme product complex, where the substrate has started to turn into two different molecules and separate from each other. And then in this last part, you've got your enzyme remaining unchanged, and the products, two separate molecules that the substrate has been turned into, will leave the enzyme. So you could see the enzyme remained unchanged, and the substrate was turned into two separate products. And going through this stage is going to lower the activation energy of a reaction. Enzymes can be affected by inhibitors, which are molecules that decrease enzyme activity, and they can be affected by activators, which are molecules that increase enzyme activity. So where you have enzymes that are raising the rate of speed of a reaction or lowering the activation energy needed, the inhibitors are going to slow that process down, and the activators are going to speed that process up. So where the enzyme is acting on a chemical reaction, these other molecules can also interfere with the chemical reaction and either speed it up or slow it down. The main thing to remember about enzymes is that these are highly selective catalysts, and they are going to increase the rate of a reaction by lowering the activation energy needed, and at the end they are going to remain unchanged. Gene mutations. Gene mutations have several different types. So one would be a missense mutation, which is where one segment of DNA is substituted for another segment of DNA, which tells the gene to make a certain amino acid. And if it's a missense mutation, then it's telling it to make the wrong amino acid. It did code for an amino acid, but the wrong one, and so this could mess up the chain. You could also have a nonsense mutation, where again a segment of DNA is substituted for the wrong segment of DNA, and it doesn't code for anything. It doesn't code for an amino acid at all, and so the chain ends there, and so it cuts that gene short and could affect the gene function. You also have insertions, which is where a segment of DNA or a base pair is inserted where it doesn't belong, and it alters the arrangement of base pairs, and it could cause the chain to end early, but it's definitely going to change what the chain does, and the function of that gene, that segment of DNA, is going to be changed. You have deletions as well, where a base pair is just deleted, and then most likely the chain is not going to function anymore, and that genetic function is going to fail. So there are different types of genetic mutations. You can have substitutions, where you just have this point substitution, this one area where a segment is changed. You can have insertions, and then you can have deletions. And sometimes this leads to just that one small incident, and sometimes it leads to replication, where there are lots and lots of mutations instead of just the one mutation. Some causes for gene mutation are environmental causes, such as chemicals that you either ingest or that touch your body, radiation, and ultraviolet light, such as from the sun. If you have too much, or sometimes just any amount of some of these things, especially chemicals, any amount of certain chemicals, and then too much radiation or ultraviolet light can make your cells mutate. And it isn't something that happens always, and it isn't something that you can say this will specifically happen, but these are environmental causes linked to gene mutation. Biological causes would be errors made during mitosis and meiosis, and these would be something that certain mutations that might occur in a child whenever the two parents are creating the child. So when that child is forming and mitosis and meiosis are going on, any kind of hereditary mutations are going to be created in that offspring at that time. So you can have environmental causes and biological causes for these gene mutations. And then results of the mutations. Well, the original course of that DNA sequence is altered. So now something else is going to happen. There could be no effect. It could just end. It could not have any effect at all if that DNA chain ends and that function isn't carried out. Maybe it wasn't something very important, and so there's no effect on the organism. it could alter the product of a gene. So the gene's product could come out differently than it was supposed to. Or it could prevent the gene from functioning properly or completely. So maybe the gene's product doesn't get shipped out at all, or it ships out something that it's not supposed to ship out, or it ships out part of something. And the results can be neutral. For instance, a gene mutation in your eye color. You can have brown eyes, blue eyes, green eyes, and the mutation doesn't harm you or benefit you. Generally, it's just going to be a difference, and so it would be a neutral mutation. You can have harmful mutations, such as that causes Down syndrome. You've got an extra chromosome there, and that mutation causes physical and mental problems. And then you can have beneficial mutations, such as that causes sickle cell disease. In the area where sickle cell disease developed, people that had the sickle cell disease were able to fight off malaria better, and so it was a survival benefit to have that mutation. So gene mutations can be caused lots of different ways, and there are lots of different types of mutations, and while most of them have little benefit or harm to us, there are some that can harm or benefit us. But genetic mutation is something that's necessary because it's what keeps people and other organisms evolving and becoming more and more advanced organisms. Mitochondria. When you see the word mitochondria, you're looking at the plural form of the word. The singular form is mitochondrion. So whenever you see those, don't be confused or think you're looking at a different word. It's just the singular and plural forms. First, let's talk about the structure of mitochondria, and then we'll talk about its functions. So the structure of a mitochondria. If you see it floating around in the cytoplasm, it kind of looks like a jelly bean. If you can see a cross cut out of it, then you're going to see this little squiggly membrane going through the middle there. So let's look at what each part of the mitochondria is. This outer layer that you can see is called the outer membrane. Next, this space in here between the outer membrane and this next line is called the intermembrane space. And then this next line is the inner membrane. So that makes sense, right? You've got your outer membrane, your inner membrane, and then the space between it is just the intermembrane space. Outer, inner membrane, intermembrane space, the space between the two. Next, you've got the space created through all these little zigzaggy patterns of the inner membrane, and the spaces that are created here and in there and down in here between where it zigs and zags, and you've got that space between each little fold. That is called the cristae. And then, last but not least, everything inside that inner membrane is called the matrix. So there you've got your structure of a mitochondrion, the basic parts of it, and it's important to know that they are found in nearly all eukaryotic cells. So that's not all, but most eukaryotic cells, and that's going to be your larger cells, the ones you find in plants and animals. And within the cell, it's going to be found in the cytoplasm. So you will see mitochondria floating around in the cytoplasm. And you could have one or you could have lots of mitochondria. It kind of depends on how big the cell is. But the basic structure is going to be outer membrane, intermembrane space, inner membrane surrounding the matrix with the cristae forming in the folds between that inner membrane. Okay, let's look at the functions. Mitochondria produce adenosine triphosphate, which is known as ATP. So if you see ATP, it's an energy source and it's called adenosine triphosphate. But a lot of times this is not going to get all typed out. You're just going to see ATP because it's a very common abbreviation in biology. Now, ATP is produced through cellular respiration, which is also known as aerobic respiration. And that's because it involves oxygen. It's basically the cell's breathing cycle where it gets to take in oxygen and use it. So aerobic respiration or cellular respiration are basically the same thing. It just whoever's typing or writing the paper or information you're looking at may use cellular or aerobic, but basically it's going to refer to the cell taking in oxygen. Now, the set of reactions that produces ATP is known as the Krebs cycle. So the mitochondria is involved in the Krebs cycle because it goes through some of those reactions to produce ATP. And the mitochondria is sometimes called cellular power plants or powerhouses because they create the cell's primary energy source, which is ATP. So since they create the primary energy source that the cell has to use, they are called cellular power plants. They also can regulate the cell's metabolism, and they are involved in cell division, cell growth, and cell death. So all of these processes require energy, and since the mitochondria is producing the primary source of energy, it makes sense that the mitochondria would be involved in a lot of these processes. It has a lot of important jobs, but the most important is going to be producing ATP because that is the primary source of energy for the cell, and the mitochondria is what produces it. So it's very important to remember production of ATP, power plant of the cell, for mitochondria, because that is what you're mainly going to see it associated with. Now, it can also do these other things, regulating metabolism, being involved in cell division, growth, and death, but main thing to remember is producing ATP. And you've got your structure, so remember outer membrane, intermembrane space, the space between the two membranes, inner membrane, cristae between those folds, and the matrix. Remember that mitochondria are going to be found in eukaryotic cells, and they're found in almost all eukaryotic cells, but not every single one. And most eukaryotic cells will have at least one, but they could have a lot of mitochondria, and you're going to find them in the cytoplasm. So just remember, if you're looking at a cell diagram, you're looking for that little jelly bean shape, and if you're thinking about what does a mitochondria do, think about producing energy, producing ATP. The mitochondria is your power plant of the cell. Mitosis. Eukaryotic cells reproduce asexually by dividing into two genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So, this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually, It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Plasma membrane. The plasma membrane is also known as the cell membrane or the cytoplasmic membrane. It separates the interior of the cell from the outside environment. So it being called the cell membrane makes sense because it surrounds the whole cell and keeps it safe from the outside environment. And the cytoplasmic membrane also makes sense because it's holding all the cytoplasm in. It's a membrane surrounding all the cytoplasm in the cell and protecting it from the outside environment. The plasma membrane controls the movement of substances into and out of the cell. So it controls what passes into the cell and what passes out of the cell. So if there are things that are waste and need to get out of the cell, the plasma membrane is controlling that. If the cell needs something, the plasma membrane is going to be what lets it in. And the plasma membrane is composed of a phospholipid bilayer and embedded proteins. Now, a phospholipid bilayer you may not have heard of before. So let's describe it. The phospholipid bilayer consists of many phospholipid molecules arranged into two oppositely facing layers. So this thing here would be your phospholipid bilayer. So bilayer because it's technically two layers of these phospholipids. And you can see how it kind of looks like it could be making a wall, and this whole thing would go all the way around a cell. Now, these top parts are phosphate groups, and these are fatty acid tails. So you've got phosphate group and fatty acid tails, which are like lipids. So you get your phospholipid bilayer because you have two layers of it. And these phosphate groups facing out bunch together and they repel some things. So they're going to repel what the plasma membrane wants repelled, and then they'll kind of open up and let through what the plasma membrane wants to let through. The plasma membrane also helps anchor the cytoskeleton to provide shape or structure to the cell. So the cell kind of looks like a big blob or a very small blob, and the cytoskeleton helps give it some shape, and the plasma membrane kind of holds that cytoskeleton in place because otherwise it doesn't have anything to anchor to hold on to keep that shape. The plasma membrane attaches to extracellular matrix and other cells to help group those cells together to form tissues. So On the inside, the plasma membrane is holding onto the cytoskeleton and helping give the cell shape. On the outside, the cell membrane is reaching out to the extracellular matrix, to what's outside of that cell, and holding onto other cells to help group them into tissues. Because you usually don't see one cell by itself. Your body is made up of lots and lots of cells, and those cells group together to form tissues, and the tissues group together to form organs, and all together your organs form up your body and make you who you are. So the plasma membrane has a big role in that. Now, something to remember is that plants, bacteria, and fungi also have a cell wall, which goes outside of the plasma membrane. And it blocks out the passage of larger molecules. So the phospholipid bilayer is going to keep out teeny tiny little molecules, but this cell wall is going to keep larger molecules from even getting close to the cell. So you might consider like a piece of fabric the kind of plasma membrane. More stuff can get through it, it's more flexible, where maybe a piece of cardboard would work for the cell wall. It's more rigid, it doesn't bend as easily, not as much is going to get through. Something can get through the cell wall, just as something can get through the plasma membrane. But the purpose is to try to keep out anything that the cell doesn't need or want in it, and to let in what the cell needs. So, plasma membrane has a few main functions you should remember. It is protecting the interior of the cell from the outside of the cell and limiting what comes in and what goes out. It is helping give shape to the cell by anchoring the cytoskeleton, and it is going to join with other cells to form tissues, because cells aren't usually going to be by themselves. So the plasma membrane is a pretty important part of the cell. It's what keeps each cell separate from the other. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now, the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So, adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together, and adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now, in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels pair together, and your two curved abbreviations pair together. So adenine with uracil and guanine with cytosine. Usually, RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So, in viruses, RNA can carry the genetic material, and a lot of times, if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code. Viruses. Viruses are tiny, non-living, biological infectious agents. So while they are not technically alive, they are biological agents, and they are infectious. They infect healthy cells. They are going to consist of either a DNA or RNA genome encased in a protein coat known as a capsid. So this capsid is protecting the viral genome until it can get where it needs to go to infect a host cell. and it can only reproduce, this is viruses, they can only reproduce by infecting and taking over a living host cell. So while they are non-living, they must take over a living host cell to be able to reproduce their viral DNA or RNA. Now, once they're inside a host cell, the virus may remain dormant for a while. So it doesn't always immediately enter the living host cell and attack. Sometimes it remains dormant for a while, but eventually it is stimulated into the active or lytic phase, and the virus inserts its own genetic material into that of the host. So the host cell is unaware of this, and the DNA that is, or RNA that is mixed in with the host DNA or RNA, it doesn't recognize that right away. So the virus is able to take control of the host's DNA by mixing with it and then taking over it. It causes the host cell to produce many viral genes and proteins. So once it gets in there, it starts saying reproduce, reproduce, make more genes and proteins. And the ones that it are making, the ones that this cell is making are going to be new viral proteins and genes instead of the normal cell's genes and proteins. And then all of these viral genes and proteins combine to form new virions or virus particles that destroy the host cell and are released to infect other cells. So what happens is, once the virus becomes active and it's in the lytic phase, it's going to insert its own genetic material into that of the host cell, so probably into the nucleus. The virus DNA or RNA is going to mix with the host DNA or RNA, take control of it, and then tell it to produce lots more of these viral genes and proteins, which combine to form new virions, and more are being made so fast that the cell can't keep up, and the cell is going to burst or lyse, which is where we get the lytic cycle or lytic phase from, because it is whenever the end result is going to be the cell lysing or bursting open. And once it bursts open, all these virions are released to infect other cells and go and repeat the process. Something to remember is that the genome of both DNA and RNA viruses can be either single or double-stranded. A lot of times a virus is going to not be too complex because the genome is only going to be 3 to 100 genes. It's not going to be very big, but it can be double-stranded if it is a more complex virus. So viruses are sneaky little things. They're going to be protected by their protein coat, the capsid, and insert their genome, their genetic material, into a host cell, take over, reproduce more viral genes and proteins, which form so many new virions that it causes the cell to burst open and send out more viruses into the host to attack other cells.
Today we're going to briefly go over an age-old philosophical question, nature versus nurture, or in other words, genetics versus environment. Are people born a certain way, or are they shaped and become a certain way based on the environment around them? Is it their nature? Are they born with it? Is it their genetics? Or is it nurture, their environment? And what we see is we look at the vast array and complex of human beings that some things are almost 100% genetic, and some things are almost 100% environment, and some things are a clear combination of the two. So sometimes it's not an either or, it's a both and. And we're just going to briefly go over some of those things related to human beings, genetics versus environment, nature versus nurture, and then the combination of the both. So when we begin then with looking at genetics, the nature side, what we are born with, what we find as scientists study this is that things like the color of our eyes, our blood type, the quality of our vision, whether we need to have it augmented or repaired or helped through glasses and things like that, our teeth, certain diseases, not all, but certain diseases that we might be prone to, heart disease, things like this. There's been a clear genetic link found that children tend to have eye color based on what they've received from their parents. Whether it's a dominant trait or a recessive trait, the information has been passed down genetically. You don't literally change someone's eye color by anything in the environment, normally speaking, unless there's some sort of disease involved or some sort of purposeful augmentation through colored contacts or who knows what. But genetically speaking, our eye color is passed down to us biologically and has nothing to do with our environment. The same with our blood type, the quality of vision we have, although the environment can affect our vision, whether it gets better or worse based on certain healthcare regulations, based on where the person is born, can affect it adversely one way or the other. But generally speaking, whether or not you're going to need glasses is not determined by your environment, but more by genetics. So there are certain things related to our bodies which we are just born with, certain proclivities to certain diseases, teeth, vision, blood type, eye color, things like that are clearly coded in our DNA and in our genes and passed down. Now there are certain things that are environmental and have nothing to do with our genetics. It's where we grow up, who we grow up around that influence us. So things like language. The language you speak is shaped entirely by what you hear as you're growing up. I have a friend who grew up in a bilingual home. His parents were, one was of Japanese descent, one was of American descent. He grew up hearing both Japanese and English spoken to him as a child. Now genetically, he wasn't programmed to know Japanese or programmed to know English. He was just genetically programmed to receive whatever language came in. Since he received both, as he grew up, for a time there, he actually meshed both languages, and others around him often had a hard time understanding what he was saying because he was freely associating and mixing both Japanese and English. Now, eventually, as he got older, he was able to separate the two languages and speak them both fluently. But the fact is, it was the environmental influence of hearing both languages that gave him that capability. So the language that you are exposed to from the time you're born growing up is, generally speaking, the language that you will speak naturally. You can also then learn other languages through school and classroom or living in another country. You pick up the language. So language is one of those things that is, a specific language is one of those things that's environmental and not genetic. Now, our ability to receive, comprehend, assimilate, process, and use language is something that's genetically inherent in us. There's some sort of matrix or lattice in the brain that allows the reception and processing and then use of a language. But what language we speak has nothing to do with our genes. If our children were born in Japan, they wouldn't be born speaking Japanese because they were born in that country, or they wouldn't be born automatically speaking English just because their parents spoke English. It's something that their environment would program into them. Along with that are things like religion and other social traits. What you grow up around socially tends to influence things like that in a person's life. So people aren't born a particular religion genetically from their parents just because their parents are. It's something that is socially around them and that they pick up. Same with other social traits. Whether they feel like they are motivated by guilt or by shame, we have different cultures that are guilt-based or shame-based. It's not something that's genetic, but something that's environmental and social. So those factors get a little more complicated, but it's clear, it's obvious. We picked examples to show that which is clearly genetic, that which is clearly environmental. Now there are some things that are actually a mix of the two, a conflation of the two. Things like our height. Now you might say, well, height would automatically fall under genetics. It falls partially under genetics. How tall you are is partially genetic. But environmental factors actually play a role in that. If your diet is not healthy, if it's suppressed, if it's missing necessary nutrients or enough protein, you may not reach your full height genetic potential. Your growth may be stunted because of environmental malnutrition and things like this. So how tall you get is a mix of both genetics and environment. The same with your weight. You may be genetically predisposed to store more fat in your body in particular places. You know, we know apples and pears. Apples stored above the waistline, pears store it below the waistline. This may be a genetic thing, in where you store fat and how much you're prone to store. But once again, environment plays a huge role. If you're malnourished, you won't store as much fat because you won't be able to. Once again, if you overeat or eat too much of certain kinds of food, you may store too much fat. So both genetics and environment play a role in things like height, weight, and then of course with diseases. You may have a genetic predisposition to a particular disease, but you can environmentally take steps to help prevent that. Through exercise and diet. You may also inadvertently not take steps to prevent that and walk right into it at an earlier age because environmentally you didn't take the steps necessary to help guard against something that's in your past genetically handed down to you. So we've gone over the whole nature-nurture debate here that's often argued in philosophy and realized that when research is done, when scientific observation is done, we find that some things are clearly genetic, some things are clearly environmental, and some things are clearly a combination of both to varying degrees. This has been just an overview of this very interesting and detailed topic. If you'd like to learn more about this or related matters, underneath this video there's a link. If you'll click on that link, it'll take you to a website that has that information, and while you're on that other website, you'll also find a link to an ebook that's ready for immediate download. Today we're going to take just a moment to go over Hick's law. Hick's law. Hick's law is related to reaction time to make choices, especially when an unanticipated stimulus is brought in. How long does it take someone to respond, to make a choice, to an unanticipated stimulus? So essentially, it's reaction time based on a stimulation. This reaction time increases based on the number of options available. So if there's only one option available and the unanticipated stimulus comes in, reaction time is small. There's only one option. But if there are multiple options, the reaction time increases. In fact, it's been found through study that the number of choices, every time the number of choices doubles, the amount of reaction time goes up by the same amount every time. So reaction increases by the exact same amount every time the number of choices doubles. Now, choice reaction time increases in proportion to the number of choices made available. This seems obvious on the surface, but the fact that it's so uniform is amazing. And this particular study, the ability to predict reaction times is amazing because of Hick's law. So that, given various parameters, someone could sit down using Hick's law and say, I know precisely how long it's going to take this person to respond to this stimulus based on the number of choices they have. Reaction time can be predicted. That's amazing. It has great application in things as divergent as basketball tactics on the court and military tactics on the battlefield. Given this input and these number of choices, it will take them this long to respond. Now think about that in terms of teaching a special forces unit. Okay, you're going to create this many distractions in these different areas. You're going to hit them all here. They have that many choices. We know it'll take them this long before they can organize a response to you that's cohesive. And that's the window of time you have to get the job done. That's pretty amazing. So anyway, Hick's law has established that the more choices they are, the greater reaction time becomes between when the stimulus arrives and when the choice is made and the person acts. If you'd like to learn more about Hick's law and things related to it, underneath this video you'll see a link. If you'll click on that link, it'll take you to the website that has that information. And while you're at that website, you'll also find a link to an ebook that's ready for immediate download.
MCAT Organic Chemistry. Hydrocarbons that contain 2 hydrogen atoms less than their corresponding alkanes are called alkenes, or we could also call these olefins. Now, they have at least one double bond and are thus unsaturated carbon compounds. Their general formula is C with subscript N, and then hydrogen with subscript 2N. So this is the general formula for all alkenes. Now, their names are derived from alkanes, simply by changing the ending from ANE to ENE. So you see propene here, which the alkane version of this is called propane. So we're just changing the ending from ANE to ENE. Now I said that hydrocarbons contain 2 hydrogen atoms less than their corresponding alkanes. So here, ethene would have 2 more hydrogen atoms if it was ethane. So starting out, ethene has 2 carbon atoms. So we can go ahead and fill in 2 here for the variable N, because we have 2 carbon atoms, so the subscript for carbon is going to be 2. Now, we can also plug in 2 for the N right here, so 2 times 2 is 4, so there's 4 hydrogen atoms in ethene. Now, there would be 6 hydrogen atoms if we were dealing with an alkane. So the molecular formula for ethene is going to be carbon with a subscript of 2 and hydrogen with a subscript of 4. So now we go to propene, which has 3 carbon atoms, and I can go ahead and fill in this whole column because they're each going to increase by 1 in terms of carbon atoms. So butene has 4 carbon atoms, pentene has 5, hexene has 6, and heptene has 7. So with propene, we can plug in 3 for the variable N for carbon and for hydrogen. So 2 times 3 is 6. So propene has 6 hydrogen atoms with the molecular formula of carbon with a subscript of 3 and hydrogen with a subscript of 6. Then we go to butene. We plug in 4 for N, so we have 2 times 4, which is 8. So the molecular formula here is C with subscript 4 and H with subscript 8. Now we can see a pattern here of the number of hydrogen atoms increasing by 2 for the number of carbon atoms increasing by 1. So here from 2 to 3 we have plus 1, but from 4 to 6 we have plus 2. Here again we have plus 1, but 6 to 8 is plus 2. So for every increase of 1 carbon atom, we have an increase of 2 in terms of hydrogen atoms. So we can infer that with pentene, since we increased the carbon atoms by 1, that we're going to increase the hydrogen atoms by 2. So if we plug in 5 for N right here, we have 2 times 5, which is 10. So our inference proves correct. So carbon has, there's 5 carbon atoms here and 10 hydrogen atoms. So I'm going to go ahead and fill in the rest of this chart. So hexene has 12 hydrogen atoms and heptene has 14 hydrogen atoms. And so their molecular formulas are going to be 6 carbon atoms, 12 hydrogen atoms, and 7 carbon atoms and 14 hydrogen atoms. Now, another pattern you may see in here is that there's twice as many hydrogen atoms as carbon atoms. So here for propene, we have 6 hydrogen atoms, and half of that is going to be 3. So that's how many carbon atoms we have. And then here we have 12 hydrogen atoms for hexene, and then 6 carbon atoms, which is half of 12. So we see 2 patterns that we can draw from this table of alkenes. So that's a look at the molecular formula of alkenes. MCAT, Organic Chemistry. Alcohols are organic compounds that have the hydroxyl group attached to carbon atoms. So methyl alcohol is the simplest of the alcohols. This is also known as methanol. And then the next higher alcohol is ethyl alcohol, which we can also call ethanol. Now, an alcohol is produced by replacing 1 hydrogen from an alkane with a hydroxyl group. So we are replacing this hydrogen with a hydroxyl group. So say we have methane, which has 1 carbon atom and 4 hydrogen atoms. We replace 1 hydrogen by the hydroxyl group, which is 1 oxygen atom and 1 hydrogen atom. So now we have this. So here we still have the same number of carbon atoms as we started with because we're not doing anything to the carbon. We're just adjusting, or we're really just adding this oxygen atom because we're taking 1 hydrogen out, but we're adding it right back in. So the total number of hydrogens doesn't change because we have 4 here, then we have 3 plus 1 is 4. So the only thing that's changing is this oxygen atom in terms of counting the atoms. So this was called methane, but now we call it methyl alcohol, or we could call it methanol. Now another example is ethane, which has 2 carbon atoms and 6 hydrogen atoms. Again, we're going to replace 1 hydrogen atom by the hydroxyl group, which is 1 oxygen atom and 1 hydrogen atom. And so now we're going to get 2 carbon atoms, 5 hydrogen atoms attached to 1 oxygen and 1 hydrogen atom. So the number of hydrogen atoms is staying the same because we start out with a subscript of 6. We have 5 hydrogen atoms here and 1 here, so that's 6 total. And then, of course, we have carbon, which has 2. We have 2 carbons, and then we have 2 carbons right here. So that's staying the same in terms of just counting them. But notice we do have a different structure here as far as the hydrogens go. We have the same number, but here the subscript's changing, so we have different chemicals or chemical compounds, or molecules really, that we're working with here. Now we are adding an oxygen atom here, and so that's where in terms of counting the atoms, that changes. So I hope you're tracking with me. What I'm trying to differentiate between just counting atoms and then actually looking at molecular structure. So the molecular structure is definitely changing here, but as far as the number of atoms go, we're staying the same with carbon and hydrogen, but we're just adding an oxygen atom. And so it originally was called ethane, so now we call it ethyl alcohol, or we can call it ethanol. And so if you're not tracking with the naming system, realize that in the IUPAC naming system, the names of alcohols are derived from the replacement of the E at the end of the word. And so it's replaced with OL. So say we had propane, we would change it to propanol. Or with ethane, we change the E here to OL to get ethanol. Or with methane, we change the E to OL to get methanol. So that's a look at the basics of alcohols. MCAT, Organic Chemistry. Hydrocarbons that have at least 2 carbon atoms connected by an unsaturated triple bond are called alkynes. Now, their general formula is carbon with subscript N, hydrogen with subscript 2N minus 2. So N right here, which is the subscript for carbon, is also plugged into the N right here in hydrogen. So you take 2 times N, then you subtract 2 from that number. Now, the names of alkynes are derived from alkanes. We're just changing the ending from ANE to YNE. So if we were dealing with alkanes, we would have propane right here. But in our case, since we're dealing with alkynes, we're changing the ending of ANE to YNE to get propene. Now, the simplest of alkanes has 2 carbon atoms, the simplest of alkynes has 2 carbon atoms connected by a triple bond, which is 1 sigma plus 2 pi bonds. So we call that ethene, and so it looks like this. Hydrogen and carbon connected by a triple bond to carbon and hydrogen. So right here we have this table that gives the names of the first 6 alkynes, which all have 1 triple bond. So we're going to take a look at the number of carbon atoms, and then from that determine the number of hydrogen atoms, then the overall molecular formula. So for ethene, I already know that it's going to have 2 carbon atoms just from prior knowledge. So 2 right here for the subscript is going to be plugged into the letter N right there. So if N right here is 2, then N right here is also going to be 2. So we take 2 times 2 is 4, minus 2 is 2. So then the molecular formula is going to be C2H2. Now we get to propene, and the number of carbon atoms is 3. So we plug in 3 for N, then we plug in 3 for N right here, so we get 2 times 3 is 6, minus 2 is 4. So the overall molecular formula is C3H4. So I'm going to go ahead and fill in all the numbers of carbon atoms. We continue, and we have 4, 5, 6, and 7. So for 4 carbon atoms, we're going to plug in 4 for N right here. 2 times 4 is 8, minus 2 is 6. So the overall molecular formula is carbon with a subscript of 4 and hydrogen with a subscript of 6. So now you're seeing a pattern here of every time we every time we increase the carbon atoms by 1, we increase the number of hydrogen atoms by 2. And so I'm going to go ahead and fill in the rest of this chart to get 8, 10, and 12, because we can infer that this trend or this pattern of increasing by 2 is going to continue. So let's go ahead and take a look at pentine. Here the number of carbon atoms is 5. So we can multiply 2 times 5 here, which is 10 minus 2 is 8, which is the number we were expecting to get. So our inference proved correct. So I'm going to go ahead and fill in the rest of this chart. But just to be careful, we need to check our work. So with hexine, we have N is 6 right here. So 2 times 6 is 12, minus 2 is 10. So that's our molecular formula. Then we come to heptine. Here, the number of carbon atoms is 7, then 2 times 7 is 14, minus 2 is 12. So that's a look at some of the alkynes. And just remember the basic formula we're dealing with here. We have carbon with a subscript of N, then hydrogen with a subscript of 2 times N minus 2. MCAT, organic chemistry. Isomers are organic compounds that have similar chemical formulas but different structures. And so they have the same chemical formulas, it's just a different structure of the atoms that are in that chemical formula. Now, for example, a molecule of butane has 4 carbon atoms and 10 hydrogen atoms. And so it can have 2 different structures. One is normal butane, which we can abbreviate to be N-butane. And then we have isobutane. And so N-butane is a straight chain structure. Then isobutane is a branched chain structure. But both of these structures contain 4 carbon atoms and 10 hydrogen atoms. And so that fits very nicely into the definition we have here because this is an organic compound that has similar chemical formulas but just different structures. And so we call these isomers. Now we come to pentane, which has the structure of 5 carbon atoms and 12 hydrogen atoms. So here we can have N-pentane, isopentane, and neopentane. So now we have 3 different structures here: N-pentane, isopentane, and neopentane. So these are all isomers of each other, because these all have similar structures, but they have, they have similar chemical formulas, but they just have different structures. And so that's some of the basics of isomers. MCAT, organic chemistry. Organic acids are made up of organic compounds that have this functional group right here: 1 carbon, 2 oxygen atoms, and 1 hydrogen atom. So this functional group we can also call a carboxyl group. And so because we call that the carboxyl group, we call organic acids carboxylic acids. Whoops. So I hope you're following me here that this functional group is called a carboxyl group, and so since organic acids are made up of these functional groups, which are called carboxyl groups, we're just going to call organic acids carboxylic acids. Now, the bonds in a carboxyl group are such that the carbon has a double bond to oxygen and a single bond to the hydroxyl group, which is oxygen and hydrogen. So it looks something like this: carbon with a double bond to oxygen with a single bond to the hydroxyl group right there. Now, the simplest carboxyl group is methanoic acid because it only has 1 carbon atom. So we could also call that formic acid. Now the next simplest is ethanoic acid, which has 2 carbon atoms, which you see here and here. And so we can also call that acetic acid. So notice the prefixes here for both of the acids, meth and eth. METH is the prefix for 1, and ETH is the prefix for 2. And so that's why formic acid has 1 carbon atom, and acetic acid has 2 carbon atoms. Now, the general formula for carboxylic acid looks like this: R to the carboxyl group, just like that. And so R represents here, R could represent methyl, which looks like that, or ethyl, which has 2 carbon atoms and 5 hydrogen atoms. So basically what this R is representing is this or something else. And so it's showing that this right here is tacked onto the front of the carboxyl group. Now, the simplest carboxyl group is just one hydrogen atom. So in that case, it looks like that. And so when we write this carboxylic acid like this, the R here is just simply representing that one hydrogen atom. So that's a look at the basics of organic acids. When carbohydrates burn, they yield carbon and water. And so that's why the name carbohydrates comes from carbon and water. Now, carbohydrates include sugars and starches. And so sugars and starches are the main source of energy that your body uses. So you can see that carbohydrates are very important. Now, there's three basic types of carbohydrates I want to go over. First being monosaccharides. It's a really long word, has a complicated spelling with two C's in a row. But more important than the spelling is what a monosaccharide is. So it's a simple sugar such as glucose, fructose, and ribose. Now, we call these sugars monomers because these sugars are monomers for more complex ones. And so notice here in monosaccharides we have the prefix mono, which means one. And then I just mentioned that these are monomers, which has that prefix again of mono meaning one. Now you may be familiar with polymers, which has the prefix of poly, which means more than one. So basically these sugars are monomers for more complex molecules. So these are single molecules that are the building blocks to be able to join together with other molecules to form polymers. So remember that because we're going to talk about that more in a little bit. So the second type of carbohydrate is a disaccharide, which is a double sugar such as sucrose, maltose, and lactose. Now, sucrose is what we would call table sugar. So it's the type of sugar you use for everyday use, for baking, coffee, that sort of thing. Now, the third type of carbohydrate is starches, which are large biological polymers that consist of straight or branched chains of monosaccharides linked together. So let's go ahead and break that down because that's kind of complicated. So these are large biological polymers. So we see that prefix of more than one. And remember, I talked about these being monomers. They're going to join together to form the polymers. And so we see that played out right here because these monomers join together to form polymers. And we even see that confirmed or spoken of again later in this definition. These polymers consist of straight or branched chains of monosaccharides linked together. So starches are basically made up of monosaccharides linked together either in straight or branched chains. Now, in complex sugars, the monosaccharide monomers combine by condensation reactions where water is removed in the form of a hydrogen atom from one monosaccharide and a hydroxyl group from an adjacent monosaccharide. So that's a look at the three types of carbohydrates. MCAT, organic chemistry. Isomers have the same chemical and molecular formulas, but they have different structural formulas. So basically they have the same chemical composition, they're just structured different ways. And so they're made up of the same chemical and molecular formulas. These chemical and molecular formulas are structured in different ways in these isomers. Now, because of their differences in the structures and therefore their electronic configurations and bondings, isomers exhibit different physical and chemical properties. Now that leads us to 3 points about isomers, or the 3 characteristics of isomers that you need to know. So the first one is isomers occur in organic compounds that have more than 3 carbon atoms. So you need to realize the word more and 3 right here. So that leads us to the second point. Organic compounds with 4 carbon atoms have 2 isomers. Those with 5 carbon atoms have 3 isomers. And those with 6 carbon atoms have 4 isomers, etc. So notice we started with 4 carbon atoms because isomers must have more than 3. And so with 4 carbon atoms, that gives us 2 different isomers. But as we increase the amount of carbon atoms, we increase the more possibilities, or we increase the isomers because we have more possibilities of structure. So finally we come to the third point, which as the number of carbon atoms increase, the ways in which the atoms can be arranged can become very complex. And this makes sense because as you have more carbon atoms, you have more possibilities as far as you can arrange them. When you only have 4 carbon atoms, you don't have a lot of different possibilities. But when you start having 5, 6, or 7 carbon atoms, you begin to have more possibilities, which makes these structures more complex. For example, you now have the possibility of straight chains with branches, or branches with subbranches. So that's increasing the complexity of these isomers. So if you had an organic compound that had 10 carbon atoms, it's going to have 75 isomers. Because you have so many carbon atoms in that case, you have so many different possibilities because of the possibilities of straight chains with branches and branches with subbranches. And so that's why you can have 75 isomers with just 10 carbon atoms. Organic compounds are basically compounds that contain carbon. There are 2 main ways to classify organic compounds. The first is by classifying where they come from. So you would have natural organic compounds and synthetic organic compounds. The second classification system is based upon how large the compounds are. So you have small molecule and polymers. In this case, we're going to take a look at the first classification system. So natural compounds are just what the name implies, compounds that can be found naturally in nature. So these natural compounds are produced by plants and animals, and humans take those compounds and use them for their own use. Sugar is a great example of a natural compound because it's naturally occurring in nature because it's produced by plants. Synthetic compounds are produced by humans, and humans produce it and then use it for their own use. Now, sometimes these synthetic compounds can actually also be produced by nature, but for whatever reason, industry has found it more efficient to produce it synthetically. A great example is rubber. Rubber can be produced naturally by plants, but industry today finds it more efficient to synthetically produce rubber. So that's a quick look at organic compounds and how they can be divided up into natural organic compounds and synthetic organic compounds. MCAT Organic Chemistry. We're going to take a look at 6 of the physical properties of alcohols, starting with physical state. So at normal room temperature, the physical state is going to be that of a liquid, and it's going to be a colorless liquid. Now, when it comes to odor, the lower members of the alcohol group are going to have a fruity smell. It's just a fact that the lower members of the alcohol group have a characteristic fruity smell. Now when it comes to density, alcohols are lighter than water. And alcohols such as methanol and ethanol are completely soluble in water. Now, on the other hand, the longer chain members of the alcohol group tend to be less soluble in water. Now, when it comes to acidic nature, alcohols are neutral liquids. So they have no effect on litmus or other acidity tests. And then finally, conductivity. Alcohols are non-ionic and non-conductive. So the reason that alcohols are non-ionic and non-conductive is because alcohols are covalently bonded compounds. So that's a look at 6 of the physical properties of alcohols. We can classify cells into 2 categories, prokaryotic and eukaryotic. So prokaryotic cells are smaller than eukaryotic cells. In addition to being smaller, they lack membrane-bound nucleuses. So they don't have a membrane-bound nucleus. Now, they contain single-stranded circular DNA molecules that free float in, you can probably guess, they free float in cytoplasm, because cytoplasm is kind of the fluid in a cell that all the other organelles in the cell float around in. So the DNA molecules free float in cytoplasm. So some different organelles that are in prokaryotic cells are ribosomes, centrioles, often one or more flagella, and cytoskeleton elements. Then we have eukaryotic cells, and so these are going to be obviously larger than prokaryotic cells. And these contain linear, double-stranded DNA combined with histone and packaged as chromosomes. And then the chromosomes are found inside a nucleus bounded by two membranes. So notice here, the eukaryotic cells do have a membrane-bound nucleus. Now, eukaryotic cells contain a variety of other membrane-bound organelles, such as mitochondria, chloroplast, endoplasmic reticula, Golgi bodies, lysosomes, and vacuoles. Now, eukaryotic ribosomes are larger and more complex than those found in prokaryotes, which is another way of saying prokaryotic cells. Now, you may be wondering, where do you find prokaryotic cells and where do you find eukaryotic cells? Well, prokaryotic cells are usually in single-celled organisms, such as bacteria, whereas eukaryotic cells are what you find in more complex organisms. It's what you see the result of on a daily basis, because plants and animals are made up of eukaryotic cells. So that's a look at prokaryotic and eukaryotic cells and the differences between them. We can describe genes as hereditary units that code for specific proteins. And these specific proteins determine how an organism looks and/or functions. So this explains why an offspring often looks like its parents. It's because we have genes, which are hereditary units, being passed down. So traits are being passed down, and that's why the organism, the offspring, is going to look and/or function similar to its parents. Now, when a specific protein is needed, the gene coding for that protein is turned on, and its DNA segment is transcribed into a complementary strand of messenger RNA. Now, we can abbreviate messenger RNA as mRNA. Now, ribosomes attach to this messenger RNA, and they also translate the mRNA, and what that does is that builds a specific protein. Now, there's a specific version of another type of RNA, which is called transfer RNA. And so, as you can probably guess, transfer RNA can be abbreviated tRNA. And so this transfer RNA is attached to specific amino acids. So the transfer RNA is attached to specific amino acids. Now, we also have something called codons, which is a term you need to be familiar with. So codons are formed of every 3 bases on the mRNA strand. So they're formed of every 3 bases on the mRNA strand, and they match up with anticodons on its intended tRNA to form a growing peptide chain of amino acids linked together by peptide bonds. So everything we just talked about can be described by this chart down here. This is a chronological chart of what we just talked about. So we have a DNA gene. Remember, don't forget why we even went into all of this. It's because a specific protein is needed. And so the gene coding for that protein is turned on, and its DNA segment is transcribed. So the DNA gene is transcribed into mRNA. Then from there, ribosomes attach and translate it to build specific protein, which we see here. And then the transfer RNA is attached to specific amino acids, which forms a growing peptide chain. And then the amino acid chain eventually becomes the peptide or protein coded for by that gene. And so from there we get the protein, because remember we said a specific protein is needed. So we had this DNA gene because the DNA coding for that protein is turned on. So we find the specific DNA gene, it's turned on, it's transcribed, and we get to that specific protein that we were trying to get to in the first place. Now it's also important to notice that in some viruses, genes are formed directly from RNA. So they don't go through the process that we just talked about, because here we started out with DNA. In that case, with some viruses, genes are going to be formed directly from RNA, not DNA. When you think of the circulatory system, you probably think of blood being moved around the body. But the circulatory system has more functions than just producing, carrying, and containing blood throughout the body. So we're going to take a look at those functions. The first is to conduct nutrients and oxygen to the tissues. The second is to remove waste materials. So this includes transporting nitrogenous compounds to the kidneys and carbon dioxide to the lungs. Those are waste materials that the body can no longer use. So by transporting nitrogenous compounds to the kidneys, it'll be excreted out of the body, and then by transporting carbon dioxide to the lungs, then the person, when they exhale, they'll breathe out that carbon dioxide. And then the third function is to transport hormones to target organs. Another name for hormones would be chemical messengers. Another function, modulate and integrate the internal milieu of the body. Also, transporting agents. And so these agents serve the body in allergic, immune, and infectious responses. The circulatory system also initiates clotting. This helps prevent blood loss. The circulatory system also helps maintain body temperature, and then the function you were waiting for, produce, carry, and contain blood. And finally, it transports body reserves to areas of need, specifically mineral salts. So that's a look at some of the functions of the circulatory system.